Okay, so I'll tell you about work that I did during my PhD in Joe Paton's lab at the Champalimau Neuroscience Institute. And so let's start with this very simplified version of the brain. So when producing behavior, the brain must combine ongoing sensory information about the environment with prior experience. But to study decision-making processes in the laboratory, psychophysical tasks have been powerful because they give us a quantitative handle onto internal variables that we think contribute to animals' decisions. But in these tasks, even highly trained animals will sometimes exhibit variable performance, or they'll fail to adhere to the rules of the game. And although typically this variability is assigned to internal states, we know relatively little about how mechanistically these internal states impact performance on these cognitively demanding tasks. And so today, I'll tell you about how task engagement might contribute to variable performance, and I'll show you evidence supporting a role of basal ganglia nuclei in representing this state. And so first, let's start by defining task engagement, because it might mean different things to different people. And if you look at the human literature, a core feature of engagement is that animals display increased allocation of attentional resources to stimuli in their environment that are related to the task at hand. And so we have been studying a particular cognitively demanding task in the lab that requires impulse control, patience, but also a difficult discrimination of an internal generated evolving variable, which is elapsed time. Sorry for the cropped plot. Okay. So in this task, rats and mice self-initiate a trial at a central nose port. And when they do that, they hear a tone, followed by one of several possible delays, after which a second tone is played. And at this point, they must choose either a left or a right nose port in order to receive reward, depending on whether the delay between these two tones was either shorter or longer than a 1.5 second category boundary. And if I plot the probability of a long choice across the stimulus duration, you see that mice discriminate the easiest stimuli very well, but choices become more variable as the stimulus duration approaches this categorical boundary. And now one hallmark of disengagement is low performance. But in this task and in others, changes in performance are due to a range of behavioral and internal, so task-related internal and external factors. And so first we built a logistic regression model to predict performance, allowing us to control for these tasks and behavioral variables that are not thought to relate to engagement. So first, let me describe these predictors. So they include the current stimulus identity, the response time, or the time it took from the second tone presentation to the choice the animal made, and also a range of other predictors that reflect aspects of motivation and vigor, such as previous reward history, when in a session a trial occurred, and also behavioral metrics before a trial even begins. So for example, the latency to initiate a trial or variability in movement. And what we found was that while current stimulus difficulty and response time were strong predictors of performance, which you expect since you just saw psychometric curves, we found that performance was much less dependent on these other variables that related with motivation or vigor. And so we cross-validated this model, and here you can see that in red, it's able to predict the main trend in the animal's performance. And then, so this model, it's predicting performance on valid trials where rodents waited for the two tones before making a choice, but sometimes animals don't follow the rules that we imposed. So on some trials, mice interrupt this side nose port a bit too soon, and we label those trials as premature. And on rare occasions, they might initiate a trial, but they fail to report a choice. And we label those trials as missed trials. Now, if we go back to the definition of engagement that I gave you before, and again, looking at human studies, we can identify behavioral signatures of engagement in this particular task. So when animals are engaged, we would expect to observe high levels of performance. And since they should be following the rules of the task, premature and missed trials should be kept at a minimum, and the opposite trend should be true for a disengaged state. And indeed, these behavioral signatures are akin to poor performance, impulsivity, and distractibility that is found in humans when disengaged. So the next question is, can we find neural signatures of such a state? And for that, we turn to the midbrain dopaminergic system, because these neurons and dopamine itself has been linked to different internal states, including deficits in attention. And so we use fiber photometry to record the activity of population of dopamine neurons at different target sites. So either at cell bodies in the SNC or VTA, and also at their projections, either in the dorsal striatum or nucleus accumbens, while animals performed this task. And here I'm plotting a single trial, calcium trace, where you can see responses to the main task events. And while in published work, we analyze this within trial dynamics and how these relate to temporal judgment, today I'll focus specifically in this 2-second period before trial initiation, where we found signals related to the state of engagement. So first, during this pretrial period, we found that dopamine neuron activity was variable, and if we bind this activity into tercels, we saw a tight correlation with performance, so that the higher the activity in purple, the lower the performance. So this was quite striking, and we think that this pretrial signal reflects a more general state of engagement, because on trials where animals fail to adhere to the rules of the task, so these premature or missed trials, just like in low performance trials, dopamine neuron activity was elevated. So we observe this inverse relationship between dopamine activity in this pretrial period engagement in both cell bodies and projection sites, which argue for a global signal that is carried throughout within these dopamine neurons. So now, if we unilaterally express TD tomato in dopaminergic neurons, and then look at the projection patterns across the brain, as was illustrated before, we can see very clearly here in this in red that one of the main targets of these dopamine neurons is the striatum and the ipsilateral striatum. So we next hypothesized that striatal neurons should also reflect signatures of engagement. And to test this idea, we again used fiber photometry, but this time we identified either medium spiny neurons or MSNs in the direct or indirect pathways of the dorsolateral striatum. And indeed, striatal MSN activity was related to performance, but interestingly now, higher pretrial MSN activity relates to better performance. So in other words, while the pretrial dopamine signal was inversely related to engagement, this MSN activity in both pathways positively correlated with engagement. So what these results suggest is that these signals are widespread across these basal ganglia nuclei, and we then wanted to know if adding these neural signals to our logistic model would provide additional explanatory power to this logistic model. And indeed, the coefficient values for these neural activity predictors have a comparable magnitude to other relevant factors. And predictions of performance were improved by adding this pretrial signal. So in all animals represented here by dots, adding this pretrial signal improved the deviance explained by the model. And we used a similar model to predict premature choices, and we observed similar results. So together with these results indicate is that this pretrial activity in the basal ganglia reflects a state of engagement above and beyond the influence that other task factors exert on decisions. Now you might wonder, how might such a signal that occurs seconds before a trial begins impact decisions that only happen later? So one hypothesis is that these pretrial levels of dopamine might determine the fidelity with which a decision variable is represented. by, for example, altering the initial state of striatal dynamics. So in previous work, our lab showed that elapsed time, which is the central decision variable in this task, can be decoded from the activity of striatal populations. And so we hypothesized that when task engagement is low and dopamine neuron activity is high, this initial state of striatal neuronal populations might be further away from the average, and this could result in outlier trajectories that wander off and therefore result in poorer representation. So as a first step to test this idea, we took advantage of single unit recordings in the striatum of rats performing this task. And we labeled trials as either typical in green or outlier in purple, depending on how similar they were to this average trajectory. And then we plotted the performance in these trial types, and we found that performance was poorer for outlier trials, which suggests that indeed the representation of this variable might be compromised in these trial types. And so just to summarize, what I showed you is that task engagement signals can be found in midbrain dopamine neurons, and in their, both at the cell body level and at the projection level, and that similar signals, albeit of opposite sign, can be found in striatal projection neurons, and that these signals provide additional explanatory power beyond what we could explain with behavioral and task-related factors, and that these task engagement signals might lead to low performance by impacting this fidelity with which decision variables are represented. And so I just want to finish by saying that these results are a first step towards understanding how circuit-level mechanisms might relate to fluctuations in engagement. And these might be particularly relevant for our understanding of conditions such as ADHD, which share similar behavioral signatures but in a more sustained manner, such as impulsivity and distractibility, and that can be alleviated by drugs that target the dopaminergic system. So for example, the amphetamines, like other reuptake inhibitors which are used to treat ADHD, have been shown to lead to a drop in dopamine neuron firing, as you can see here. And we think that it's possible that these results mimic more naturally occurring states of engagement, such as the ones that I showed you today. And so I would like to finish by acknowledging my PhD mentor, Joe Paton, for his support and guidance, and the whole Paton lab, in particular, Bassam, Atallah, Asma, Motiwala, Bruno, Cruz, Tiago, Monteiro, and Thiago, Gouvea for collaborating in this project. Everyone at the Champalimau Neuroscience Program, the funding sources, and you for listening. Thank you.
Thank you. We have time for one or two questions.
That was a great talk. I was wondering, in the low, medium, high situations when you showed the worsening of the psychometric functions, does that affect the slope of the psychometric functions, or does that affect the asymptotes?
Right, so I didn't go too deep into that. So we did model comparison for all those plots. So in some cases, you only see slope changes. In more extreme cases, you'll see the parameters for the lapse is also included in the model that best fits the data. So,
thank you.
So you showed correlation between pre-trial dopamine and task engagement, but I wonder whether that level is correlated, or subsequent phasic response rather more correlate to the engagement. Did you try? So if the baseline is low, you might have higher phasic response, and I wonder which one correlates better to task engagement.
So yeah, so if I understand the question, for example, if I now split my performance based on this amplitude of the first tone response, do I see something like this? And the answer is no. And I also didn't show that the autocorrelation of this pretrial signal is relatively low. So although on average you see those big changes, if you look on single trials, the pattern is not that one. So yeah.
Thanks.
Let's thank the speaker again.
Thank you.
So we're going to briefly switch speakers while we solve a small technical problem. So first, we will hear from Kristen Delevich from UC Berkeley, who will tell us about her work on the striatum.